And I want to share the ebook. So last week it was unknown for group 1 and 2. The unknown for group 1 and 2, I don't have the exact detail of the video, but I can explain using the flowchart, unless you want me to look at the specific part of the video, the recording, or if you have questions about the specific part of the recording, you let me know. Okay. Let me. Okay. We started with the unknown and added HCl. And what happened when we add HCl? Did you have precipitate or you had no precipitate?
There was white precipitate.
There was white precipitate. And the centrifuge separated, using the centrifuge, we separate the white precipitate, which could have been lead chloride and silver chloride mix, or one of them at least, plus the solution that could contain copper and bismuth, or at least one. To the precipitate, hot water was added, and when you add hot water, you can only say if you have, you could have lead, but if there is any precipitate left, definitely is silver chloride. Did you have any precipitate left after adding hot water?
No, there was no.
No precipitate was left after adding hot water. That means there is no silver chloride. So as a result, silver is absent. Now, with the lead, at that point, it's kind of confirmed, but you have to confirm it again to make sure that you have evidence. When you add potassium chromate, a yellow precipitate, it should have appeared, right? So you had yellow precipitate. Let me see. Look at the chat. There was some precipitate from what I saw, okay, and some precipitate dissolved and some was left. So you had precipitate after adding hot water. You had no precipitate left, right?
Yes.
Okay. So if there was no precipitate left after adding hot water, the solution, it would be, okay, the solution would have been Pb2 plus, and when you add the potassium chromate, you had that yellow color precipitate, right? Okay, so that confirms that you had lead. And was ammonia, did I continue with that little precipitate that you said that there was some left, or I did?
Yes, you did the whole process.
And I add ammonia?
Yes, you did.
After the ammonia, I added the nitric acid?
Yes.
Yes. And did I get a precipitate?
No.
No precipitate, okay.
No.
So there was no, if I didn't get precipitate and I said there's no precipitate, that means there was no silver chloride.
So a quick question, Professor, I do have about that. The leftover precipitate we did have, was that leftover a silver chloride that was just, that didn't dissolve in the hot water?
It could have been some lead chloride, yes, undissolved lead chloride, because depends on the temperature and the amount of the water you're adding. And by the time you do centrifuge, the centrifuge can, it can again lower the temperature. So it depends on how long it takes and how hot was the temperature or how long you had it in the hot water, and it can get, precipitate again. It can dissolve and precipitate again. So that's a possibility, yes. So leftover of the PbCl2, undissolved PbCl2. Okay, and then to the solution, we had the copper and bismuth, added sulfuric acid. With the sulfuric acid, I don't remember having precipitate, but if you had any precipitate, you centrifuge. That's just to remove if there is any leftover of the lead chloride. To the solution, then we add ammonia. When you add ammonia, bismuth would precipitate in form of the bismuth hydroxide, but copper would stay in the solution. Did you have a precipitate? No precipitate formed? Okay. And what does it mean if there is no precipitate formed there? No bismuth. Okay. So to the solution, I added the acetic acid, and continued by the K4FeCN, so potassium ferrocyanate. And you had that maroon color formed? Yes? Okay. That means copper was present, bismuth was absent, and lead was present. But do you have a question about any theory, or what is the question about last experiment? Do you still have questions about the last experiment, or should we go to the next experiment? Okay, next experiment. Next experiment is about week 3. For group 3, this is known samples again. With the known sample, you could have iron, chromium, 3+, nickel, manganese, or zinc. So there's a possibility of, actually for the known samples, so you have all 5 of them. For unknown, you could have that, which is next week, you could have 1, 2, 3, 4, or all 5 of them, depends on the unknown. But since I think I just did one unknown, you would write your report based on that unknown that was performed. So the flowchart is not the only piece of information you have to study about this experiment. You should basically read the whole experiment before you watch the video or you answered your pre-lab questions. But if you look at the flowchart, it kind of summarizes the selective precipitation process. That's your first step. So you have to find the reagent to make sure that some of these ions are going to precipitate, but the precipitate then should not be like a very stable, very strong. So because it's a sodium hydroxide precipitation, maybe later on when you add acid, you can take it back into the solution again. So it's like you neutralize the sodium hydroxide, then you release the ions, then you can separate them further. But at least you can divide into 2 groups, a group that would precipitate with hydroxide and group that do not precipitate with hydroxide. So out of the 5, zinc and chromium do not precipitate with hydroxide, so they will stay in the solution format. But iron, nickel, and manganese is going to precipitate. So you have a precipitate, which when you centrifuge, you separate into 2 separate test tubes. To the precipitate, you add nitric acid. Nitric acid, basically what it does, it neutralizes the base, and it can change all 3 of them back to aqueous solution. So basically you are removing the hydroxide from all of these 3 ions. So when you have the 3 in the aqueous solution form, they do not interfere with the conformation of each other or presence of each other or precipitate of each other, because when you divide this to 3 separate aliquots and you place it in 3 separate test tubes, you're adding the reagents, like this reagent or the color of permanganate. It's so strong, the purple color of permanganate is so strong that presence of nickel and iron is not going to affect the evidence of this reaction. So we are looking for is a qualitative analysis, and we are just trying to for evidence of the reaction. However, when you watch the video, you have to, or if you were doing this experiment, as soon as you add the sodium bismutate, the solid compound, you should look for purple color. If you leave it alone, that permanganate can be reduced to manganese dioxide, and especially with the presence of the other 2, this compound, the final product, it can go reverse. It can go to reduction and changes to manganese dioxide. And if it changes to manganese dioxide, you have brown color. And with the brown color, it's very hard to recognize if that is the manganese or if it came from iron. So you want to look for the evidence right away. But if that purple color appears, it is confirmation test for presence of manganese ion. But if the purple color does not appear, other precipitates appear, it's absence of manganese. So when you're adding sodium bismutate, your concentration is only for formation of the permanganate with the purple color. That's the purpose of this test. And for the iron, potassium thiocyanide with the iron can form a bloody red color. So it's like, it looks like basically blood is the very strong red color. As soon as you see that, with thiocyanate only iron would give that color, and the other 2 compounds could give a compound, but it's not going to give that color. So when you see the color of the bloody red color, that means you have iron. To the 3rd aliquot, you are going to add the ammonia. When you add ammonia, basically it's going to keep the nickel in the aqueous solution form. And when you add ammonia, the other 2 could precipitate. So we look at the procedure for 3E. Let's see, if you're adding ammonia, what's going to happen? You get a precipitate and the aqueous solution or not. But whatever is the aqueous solution, that is going to be nickel. We know that nickel will not precipitate in presence of ammonia. If you add hydroxide, yes, hydroxide will precipitate nickel, but ammonia does not precipitate nickel. Again, it's selective precipitation. Ammonia, in water, it produces ammonium hydroxide. The hydroxide, it can, just like the last experiment, when you add ammonia, then bismuth, it changed to bismuth hydroxide. In this case, ammonia can give hydroxide to iron and manganese, but not nickel. So the only part that we are interested on is the nickel part that stays in the solution. And to that solution, when you add the reagent of DMG, is going to give you like very bright strawberry red color. And when you see that strawberry red color, that means the nickel was present. Now, if the nickel is not present, you might see precipitate, but that precipitate is not nickel. So not every precipitate is nickel in this case. If it's a strawberry red color, then it is nickel. Okay, any questions on the left side of the flowchart? No questions? On the right side of the flowchart, what is the purpose of adding hydrogen peroxide? What will happen when you add hydrogen peroxide? Yes. Yeah.
It oxidizes the CR hydroxide, the chromium 4 hydroxide, I think is the name, into CrO4.
Mm-hmm. So chromium hydroxide will be oxidized further. So if you remember determining oxidation number in CrOH4 minus, the charge for chromium is 3 plus. When you are adding H2O2, the 3 plus is going to change to a 6 plus. So basically, chromium in chromate ion has a charge of 6 plus. That means chromium has been oxidized. So you are going to oxidize the chromium to chromate ion. And when it's oxidized to chromate ion, then with the barium chloride, it can change to barium chromate, which is a yellow color precipitate, or the tan color precipitate that you see there. So you are looking for oxidation of the chromium. Now, the hydrogen peroxide, because is an oxidizing agent, is a strong oxidizing agent, at the same time is not very stable. So because it's not very stable, it has to stay in the brown color glass container. It should be away from heat and light. And the 3% of that they're selling at the grocery stores, like the drugstores, they keep it in the brown plastic container. The one in the lab is a 30%, is a stronger, but still over the time it can lose the strength because it can decompose and change to water and oxygen. So if it decomposes, when you are adding H2O2, and especially if you pour, like if I'm just by myself, I don't pour it until last minute into the beaker. But if it's left open in beaker for like even half an hour, the strength is going to fade. So it would be weaker oxidizing agent, and it may not give high concentration of chromate ion. And when you add barium, you might get like less precipitate or less obvious color of the precipitate. So the color may not match exactly what's in the lab, in the ebook, but that's why we are performing known sample first, so you can record your numbers, you can record the observation. So I want you to record the observation that you actually see in the recording, in the recorded video. Do not depend, do not avoid watching the video, and make sure when you're watching, you do not fast forward, record your numbers, your observation carefully, because if you record your observation carefully, you will have much easier time next week when you are trying to identify your unknown. just like bismuth, when we had like a dot, black dots rather than black precipitate, in a white precipitate with the black dots, something like that could happen. So this color of the barium chromate, it can go anywhere from light, tan color to dark color. So you have to record, because if you have chromium as part of the unknown for the next class, is going to behave similarly to what your compound, your sample behaved today. Okay. How do you confirm presence of zinc? See, the purpose of adding HCl here is to get the zinc again free from this complex ion. So you want to make sure that your zinc OH4 2- is going to change to a zinc 2+. And how do you confirm you have zinc? What color would be the precipitate? Okay, let me go to the chapter so I could see. It's a gray-white precipitate, okay, a tan-yellow precipitate. So what are you going to record in your mini lab report is the color of the precipitate that you see when you add potassium ferrocyanide. Cyanide. So that's what you are going to record. In the ebook, it might be a different color than what you actually see in the lab. So your answers, it looks like that you do have some combination. Some is coming from the book, some is coming from the video, but what it needs to be recorded in your data sheet, in your mini lab report, is based on the video, please, to make sure that it does match with the video.
Professor,
Yes.
on the lab report, it says both gray-white precipitate forms confirms the presence of zinc, and then tan-yellow precipitate form confirms the presence of zinc. So we would just go by the video, correct?
Yes. You have to go with the video, because when we have that mixed color, and depends on, it might say that depends on presence of other ions. If you have another ion like leftover of, let's say, iron, it can change the color. So we try to keep the effect of the other ions consistent for the unknown compared to the known sample as well. So anything that is contributing is going to be the same, and as long as we keep it the same, we can do the identification, the qualitative analysis. Okay. Questions? Okay. All the reactions are presented here. So please do not just go with the qualitative analysis. Pay attention to these reactions. Now, what would be the reaction of the zinc with the hydroxide, or iron with the hydroxide? Even though that flowchart is going to summarize it all, but still you want to know the reaction on each step, what is happening here. Like when you have oxidation of chromium going to chromate ion, this is a chromium 3 going to chromate, which is the chromium 6+. And the yellow color is coming from the chromium 6+, but it does indicates presence of chromium 3+, because at the beginning you had the chromium 3+. With the H2O2, it changed to the chromium 6+. And when you are adding H+ or hydrogen, HCl is going to remove the hydroxide, and it makes these three again free ions. So they're not in precipitate. Again, it dissolves. Questions? Do you have any questions for me? No questions at all? Okay. Okay. So if there is no iron, you will see like tan yellow color. That could give you the presence of the zinc. But if there is iron present, then the precipitate is going to change color and the bluish tint color. So it depends on what other impurity leftover of the other ions can affect the color. So we go with the video, the color of the video, presented or evident. Questions? No questions? Okay, if you don't have questions, I'm going to call for attendance. So you can go ahead and start watching the video. Okay. For February. Megan?
Here.
Thank you. Taziana?
Here.
Juan? Juan? Absent? Saima?
Here.
Crystal? Crystal S? Okay. I think I have you two places. Okay. Robert? Okay. Natalie? Elaine?
Here.
Thank you. Janelle?
Here.
Caitlin? Isabella?
Here.
Nia? Nia?
Here.
Andre? Camilla?
Here.
Thank you. Kaylin?
Here.
Thank you. Jonica? Neftali? Okay. Alex?
Here.
Alana? Naveed? Daniela?
Here.
Yeah. Luke?
Here.
Nia? Sabrina? And Euben? Okay. So we have Nia and Naya, both of them, right? Naya? Did I mispronounce your name first? Nia and Naya, right?
Yeah,
Is that right?
Naya.
Both of them are Nia or one is Naya? You're Naya, right?
Yeah, Naya.
Naya, okay. Yes, I got it. I just mispronounced. Okay. Very good. Questions for me? No questions? Okay. If you have any questions, you may stay. If you don't have questions, please go ahead and start the watching the video. Make sure you read the experiment, watch the video, record your observation, and make sure your observations are written. You have a recorded observation. This way, when you are doing the unknown sample next week, you can refer to your recorded observation and answer the questions for that. Yes, Alex.
I didn't. So for next Tuesday, it's the post lab and the pre lab due.
The post-lab for this experiment, post-lab questions for this experiment, and a mini lab report, and pre-lab for the next part, pre-lab for the unknown.
Okay, thank you.
You're welcome. Thank you so much. Thank you. If you have questions, you may stay. If you don't have questions, you may start watching the video, please.